I want to introduce you to my friend, Roberto. Roberto is one of those kindest, warm-hearted, dedicated scientists you'll ever meet. I had the privilege of being a friend and a colleague to him. He grew up in Argentina, studied at University of Buenos Aires, and spent a lot of his career in the United States, where we met. By the time I met him, he had already had a long list of accomplishments, having helped start a company, having helped lead scientific teams with multiple patents, and really contributing to cancer diagnostics and therapeutics. And I was really, really glad to be able to work with him. And you wouldn't hear any of this from Roberto himself. He was super humble and never took credit, and was the last to say anything bad about anybody else, and the last to say anything good about himself. So I had a great privilege of working with him as we fought together to figure out how to detect cancer early using a blood test. This picture here is when we were celebrating in London. We had a major breakthrough in our collaborators in England, and so we went and visited. Since it was one of our first times to the UK, we decided to partake in the afternoon tea, and we took this picture to celebrate. About a year before this picture was taken, Roberto felt ill a little bit and went to the emergency room. And then the doctor saw something in his GI tract but didn't know what to do, decided to keep him in the hospital to study and to observe. They were able to do some procedures, and they didn't really find anything significant, so he left after a few days. About a month after we took this picture, Roberto again was not feeling well, and he went back to the emergency room. And unfortunately, this day was much, much longer. They found cancer in his body, the cancer that they didn't find the last time, and it was already advanced. And after a few weeks, Roberto passed away. We were working on the very technology that would have helped Roberto, finding cancer early so we can treat it. And the sad thing is that this story isn't unique and isn't rare. Around the world, over 9 million people die of cancer every year. That's 3 times the population of Quito. Imagine, every year, 3 times the population of Quito dying from this horrible disease. And then but you hear in the news that there seems to be a new cancer cure. Almost every day you sort of open the newspaper or you surf on the internet, there seems to be a lot of great news about cancer. So how do we sort of reconcile the paradox of all these amazing discoveries, but more and more cancer cases coming? And even more, the WHO, the World Health Organization, estimates that by the year 2020, that number, 9 million, will increase by another 70%. This word often, when we hear the word cancer, strikes fear and anger and sadness in all of us. But I want to give you a quick overview of what's been happening in the cancer field and give you a sense of hope. What's happening in cancer? What's happening in the front lines of cancer? And is there a solution potentially to fight this horrible disease? So in fact, we are living, for those who aren't knee-deep in cancer research, we don't understand that we're actually in the middle of 3 of the most exciting revolutions in cancer research. The first one is cancer genomics. The genome is the entire DNA, all the genetic material of an organism. Changes of a DNA, called mutations, are ultimately what drives cancers to grow and proliferate. About 10 years ago, I was part of the team at Johns Hopkins in Maryland, Baltimore, Maryland, in the US, that we were one of the first teams to decode comprehensively the mutations of cancers. We did it first in colorectal and breast cancers, then pancreatic cancers and brain cancers. Today, there are over 90 projects looking at all different cancers in 17 countries, working hard to sort of figure out the genetic basis of cancers, and we're making a lot of progress. Tens of thousands of cancers are now understood to intricate molecular detail. We know many of the causes of cancer, and we know how to start targeting them. We have the map. The second big revolution is precision medicine, also known as personalized medicine. So originally, when cancer was being treated, it was given sort of these one-size-fits-all, often derived from very toxic chemicals, as sort of chemotherapy. Some of them have derived from mustard gas. And the key is to kill the cancer without killing the person. But with these recent sort of developments in understanding the genetics of cancer, scientists have been able to develop drugs that target those mutations only in the cancer and not in the normal person. And this is called precision medicine. So then every person, after you map out what's the genetic mutations in that cancer, you can give different treatments to different people. People often say this is the right drug to the right patient at the right time. In the last 10 years, there are scores of new targeted medicines now available for oncologists to be able to custom tailor drugs for cancer patients. The third and recently most exciting development is immunotherapy. So instead of giving sort of drugs, what scientists have been able to do is to leverage our own immune system, which already fights diseases and fights cancers in our body, and be able to leverage it to be able to fight disease. For example, scientists have been able to find an off switch of the immune system and actually develop drugs to turn it back on. And already, some of them being clinically approved and being used by people all over the world. There's even more high tech where people are able to take immune cells from the body, sort of engineer it, train it in different ways, and then put it back into the body to fight cancer. Some of this sounds almost like science fiction, but it's happening right now. And it's shown a lot of promise. This area of immunotherapy is one of the most promising areas of cancer therapy. While I was a researcher at the National Institute of Health in the US, at the National Cancer Institute, I was able to work with some of the leaders and pioneers of this field, and I've been able to see the explosion of this area of research. Today, there's hundreds of trials now available for patients to participate in, to understand and be able to develop these immunotherapies. So these are the three of the most exciting areas and revolution, completely new ways of addressing cancer. But why is it that still people are suffering from cancer today? Let me illustrate with one patient. So this is a patient with this cancer called melanoma. It's a skin cancer. It looks horrible. The cancer has grown everywhere in his body. However, scientists were able to sequence the genetic material of this cancer and find that there's a specific mutation where we have the drug to. So they gave this drug to this patient, and this is what happens. It's amazing. The tumor melts away. You're like, wow, this is amazing. This is the future. The sad thing is, this is not the end of the story. A few months later, this picture is taken. The cancer has come back. The cancer has learned how to evade the drug. So the question is why, and how can we fight this? The answer is this term called tumor heterogeneity. Let me explain. So in the cancer cell, let's say as small as 1 centimeter in size, so this is about as small as this, 1 centimeter, there's over 100 million cells and up to 1 billion different cells, all different cells. And even though that they're mostly the same within these cells, there are small genetic differences that make them respond to drugs differently. So if you even have a drug that's able to kill off almost all the cells, right, there could be a small population of cells that doesn't respond to drug. And this ultimately is the area that has resistance and takes over the body. So the key to curing cancer is to use all these sort of amazing therapies and interventions that we have early before their cancers have grown large enough so that there's resistance clones to be available. Ultimately, the key to curing cancer is early detection. And intuitively, we know that. We sort of know if you catch cancer early, people will do better. And the numbers show that as well. For example, in ovarian cancer. In ovarian cancer, if the cancer is caught early, for example, in stage 1, 17 out of 100 patients are able to survive at 5 years. Wow, that's actually pretty good. However, if it's stage 4, sorry, let me just correct that. So in ovarian cancer, if you catch the cancer in stage 4, only 17 women out of 100 will survive. So most of the women die at 5 years. However, if you're able to catch the cancer in stage 1, 92% of the women will survive. Right? So the key then is to catch ovarian cancer early and catch these cancers early, because even with the drugs that we have today, people will already do better by catching them in an earlier stage. But the sad fact for ovarian cancer is only 15% of women are caught in stage 1, whereas the vast majority, 70%, are caught in stages 3 and 4. So the key is figuring out ways to be able to detect cancer early and be able to apply. So what are the current methods to detect cancer early and to be able to do this? The first one here is a medical procedure. So for example, colonoscopy for colon cancer. It's a medical procedure to put in a tube to be able to see whether there's cancer. Second one is using protein biomarkers, for example, PSA for prostate cancer. The third one is imaging, sort of taking pictures and x-rays and CT scans. The most famous one is mammography for breast cancer. Medical procedures, while they are the gold standard, they're great. The thing is, they're highly invasive. These procedures, you need to do a lot to be able to do. And it's infrastructure intensive. You need a big hospital, you need doctors, you need a prep to be able to do that. Protein markers, on the other hand, is potentially easy to do, but it's low specificity, meaning that there's a lot of false positives. So with a false positive, then people are scared. They think that they have cancer, but they don't, and they result in unnecessary workups. The third is imaging. Imaging, great for some uses, struggle from radiation. You don't want to use it too much. You can't do it very often, right? Because you're exposing to harmful radiation, which ultimately causes cancer. And then even more, it's not applicable to all populations. For example, women with dense breasts. We need a method that is non-invasive, that is very light in infrastructure, that's highly specific. What we need is a method that is non-invasive, that is very light in infrastructure, that is highly specific, does not use harmful radiation, and ultimately is applicable to all populations. Does such a technology exist? Well, I wouldn't have flown all the way from the US if it doesn't. Us at a company, Natera, we've been working on this technology. I'm happy to sort of tell you more about it. So central to this technology is a simple blood test. The blood circulatory system, while seemingly mundane, is very, very important in sort of sustaining your life. It provides oxygen and nutrients to your cells and takes away carbon dioxide and waste. Here's the key. Cancer cells grow and die faster than normal cells. And when they die, they shed off DNA, which can be seen in the blood. And from the Cancer Genome Projects, we know the signatures of these cancers, so then we can look in the blood for these specific signatures in order to detect cancer early. So instead of waiting for cancers to be large enough so you can visualize it on a medical procedure, or dense enough so you can see it on imaging, or significant enough that it causes symptoms, we can start looking for cancers while it's still in very small amounts by looking for small amounts of DNA in the blood. So how do we do this? So the method starts off actually pretty simple. We start off with a simple blood draw. No special material, no special procedure, a simple blood draw. Then we separate out the cells from the blood and look at the DNA in the plasma fraction. Most of the DNA that actually comes from your body is normal, and only a small amount of it comes from the cancer. The key is to be able to find that small amount. Then we use a series of molecular biology techniques to focus on the areas of DNA that we know are important for the signatures of cancer. Then we take the DNA that we've prepared, put it in a DNA sequencer, and is able to read out and digitize these DNA into letters A's, C's, T's, and G's. Ultimately, the output is billions of letters of A's, C's, T's, and G's, and we use computational and statistical methods to be able to find a small amount of signal in the noise. Does this work? Yes. And in fact, we're very excited that we just published this data very recently and was actually featured on the cover of Nature magazine, which is one of the top scientific magazines in our work. We started this by doing it in lung cancer because we can not right now predict who's going to get cancer yet. We started in a population that's second best, is focusing on populations in remission. So the sad fact is, despite all that technologies that we have, lung cancer, after you treat it, ultimately comes back. So the key then is for us to measure the DNA in these patients under remission and see whether we can catch these recurrences early. Working with Cancer Research UK, University College London, led by Dr. Charlie Swanton, we just celebrated in the UK, we were able to publish this result. So let me walk you an example of one patient. So this patient starts off with surgery at day 0 and ultimately has chemotherapy given. And then there's a time of waiting to see, this patient supposedly doesn't have any cancer, and is it ultimately going to come back? Methods is imaging or clinical visits. And ultimately, unfortunately, around day 450, the cancer comes back. During this whole time, we've been measuring blood serially. And we can then ask whether is there DNA in the blood that we can look at. So in the beginning, when it's during surgery, there's a high level of DNA from the cancer in the blood, which then goes to a very low amount through subsequent time points. And ultimately, around day 340, there's a rise that we see in the amount of cancer DNA in the blood, eventually going up to higher and higher amounts. The key here is we are detecting the rise of cancer DNA at day about 350. This is about 100 days earlier than clinical detection. This is 100 days potentially earlier that you can give treatments or therapies. And more importantly, this is 100 days less for the cancer to grow resistant clones. And oftentimes, 100 days for some cancer patients can mean the difference between life and death. We've been really excited about this research. We did it in the first 100 patients, 24 of them, of which we did longitudinally. We achieved a sensitivity of 93%, a specificity of 100%, and to some patients, we're catching these cancers up to a year in advance. Excited by all this data, we started working in sort of later data to be able to see in other cancer types as well as in lung cancer. We're looking at breast cancer, we're looking at lung cancer, and we're looking at ovarian cancer, and I can't wait to see how much earlier we can catch these cancers. Ultimately, I have a dream of 2 tubes of blood. That as part of your annual visit to your doctor, you'll have 2 tubes of blood drawn, additional plasma drawn, and using these 2 tubes of blood, we'll be able to sequence all the DNA and compare it to all known cancer signatures, potentially finding cancers days, months, and potentially years in advance. Even with the therapies that we have today, this could mean that millions of lives can be saved. This is the dream for patients like Roberto. If this were available to him, potentially he would be with us today. So the next time when you hear the word cancer, I wanted to add to your emotions of sadness and fear, the emotion of hope. Hang on. Millions of researchers around the world are making enormous strides in cancer research. And we're starting to see the beginning of the end. The end is near. We will win the war on cancer. Thank you.